right, so we're going to pick up where we left off talking about different organelles of a eukaryotic cell. Okay, we went over the parts of a prokaryotic cell. Now we're focused on eukaryotic cell. Okay, and one of the characteristics of a eukaryotic cell is that it has membrane-bound organelles. So we talked about the nucleus first, different parts of the nucleus. Now what we're going to go to is first going to talk about the endomembrane system, and then go into some other organelles that are highlighted here, some that are specifically to the plant cell, some that are specific to animal cell, and some that they both have. All right, and the one we're going to start with, endomembrane system, they both plant and animal cells have. All right, so endomembrane system has three functions. It interacts between the nucleus and the plasma membrane. It produces important molecules for cellular function, and it works to destroy toxins, recycle waste, etc. Okay, and the first part that we'll talk about is endoplasmic reticulum. There are two types of endoplasmic reticulum, which is an organelle formed of continuous sacs and tubes. So you have the rough endoplasmic reticulum. All right, the reason why it's called rough is because they have ribosomes attached to the surface of them. So here you see these tiny ball-like structures. Those are the ribosomes. So rough endoplasmic reticulum has ribosomes attached to the surface. Proteins are synthesized at the ribosomes and then enter the rough ER where the protein structure is finalized. All right, then you have the smooth ER. Okay. All right, so you have the rough ER here and the smooth ER here. Now, as you can see, they have a direct connection to the nucleus, all right, that you see here. The smooth endoplasmic reticulum does not have ribosomes attached to it. That's why it has a smooth appearance. But some of the proteins finished in the rough ER play a role in the smooth ER as enzymes. Okay, and we talked about enzymes. Most enzymes are proteins, that class of organic macromolecule. The smooth ER is also the site of lipid formation and digestion of other macromolecules, as well as other toxins. All right, so moving on to vesicles, okay, which are still considered part of the endomembrane system. All right, so vesicles are membrane-bound organelles whose main function is to transport a variety of substances to and from organelles and out of the cell. Okay, so transporting, collecting, and disposal. Three important functions of vesicles. Some examples of vesicles are lysosomes, okay, and peroxisomes. So vesicles can form by budding from the plasma membrane or by budding from other organelles. All right, vesicles that bud from the endoplasmic reticulum are called peroxisomes, and they break down fatty acids, amino acids, and toxins. Okay, vesicles that bud from Golgi bodies are called lysosomes, and they have very powerful enzymes that break down organic molecules. All right, so as you can see here, all right, you have parts of the Golgi body or Golgi complex budding off to become lysosome vesicles. All right. Then you have the Golgi bodies. All right, they are organelles that resemble a stack of pancakes. All right, seen here. So the Golgi bodies are termed the packaging center of the cell because this organelle has enzymes that finalize the proteins and lipids created in the endoplasmic reticulum and package them to be transported to lysosomes or the plasma membrane. All right, then one last type of vesicle are vacuoles. Vacuoles are fused vesicles. Okay, they're vesicles that have fused together. All right, and the vacuoles function to isolate or dispose of various substances. Okay, so they isolate and dispose of waste and toxins. Now you have a specialized vacuole in plants called the central vacuole. All right, animal cells don't have a central vacuole. So the central vacuole in plants takes up a large part of the cell, and the fluid in the central vacuole gives the plant cells the necessary pressure so the plant cell can keep its overall form. All right. So a large part of the water that a plant cell takes up will go into the central vacuole, and we'll talk about that more in the next lecture. Okay, next you have mitochondria. Okay, now mitochondria are found in both plant and ant cell. Okay, mitochondria are double membrane, so they have an inner and outer membrane, and they are nicknamed the powerhouse of the cell because that is where ATP is produced. And ATP is a major energy molecule for the cell. Now, if the respiration happens in the presence of oxygen, you have aerobic respiration. Okay, and most respiration that occurs in a cell is, or in different organisms, is aerobic respiration. All right, the number of mitochondria can vary per cell type. Those cells that need a lot of oxygen, that need a lot of energy, are going to have more mitochondria. And a good example of this are your muscle cells. Your muscle cells have a large number of mitochondria because they use up a lot of energy. They use up a lot of ATP. You have the different parts you have, like I said, the inner and outer membrane. Then you have the folding of the inner membrane, and those are called cristae. And then you have a fluid inside the inner membrane called the matrix, mitochondrial matrix. Okay. Now, it's scientists have looked at how cells first derived their mitochondria. How did they sort of produce these mitochondria? How did they come across making these or having these internally? And what scientists have come up with is this endosymbiont hypothesis. Okay. The reason why they came up with this hypothesis, basically saying that millions of years ago, okay, a cell basically took up an independent cell, all right, and then the independent cell eventually, over time, became mitochondria, all right, that would produce the energy for the cell. But how did they come up with this idea that the mitochondria were actually independent cells? Well, mitochondria have their own DNA. They have their own ribosomes. Okay, so that supported this idea that mitochondria were actually, before they became part of the cell, they were actually derived from unique independent cells because they have their own DNA, they have their own ribosomes, and plus they divide independently of the cell which houses them. All right, and because of those characteristics, scientists theorize that mitochondria evolved from independent aerobic bacteria that took up residence in eukaryotic cells. All right. Before I get off mitochondria, another interesting aspect of mitochondria is that they are maternally inherited, meaning all of the mitochondria you have in your body were given to you from your mother. You didn't get any mitochondria from your father. They only came from your mother. All right, another type of membrane-bound organelle are plastids. Plastids are organelles that do not occur in animal cells, and they function in photosynthesis and storage. Whether it is pigment storage or food storage. Okay, plasts like mitochondria are double membraned. All right, and the first one we'll talk about are chloroplasts. Now, chloroplasts are plasts that specialize in photosynthesis, and they store a photosynthetic pigment, the main one being chlorophyll. All right, and that's why plants have their overall green color, because they store this green pigment, chlorophyll. Now, scientists also think the same way cells derived mitochondria or evolved to have mitochondria, they also theorize the same endosymbiont hypothesis for chloroplasts, because, again, chloroplasts have their own DNA. So millions of years ago, a photosynthetic organism was basically taken in by another cell, and it basically became a part of the cell over time. All right, so here's a photo, and all these green circles are chloroplasts that are housed within these squared plant cells. All right, other type of plastids, you have chromoplasts that create and store pigments, just not chlorophyll. Okay, so they store pigments, they produce pigments, just not chlorophyll. So you have the pigments can be orange carotenoids, yellow xanthophylls. Well, what's the purpose of these pigments? Well, some of it, they can absorb sunlight, but they also can give the plant or the fruit color to attract pollinators. All right, and then another type of plastids are leucoplasts. They store carbohydrates, lipids, and proteins. They're colorless, and a good example are amylioplasts that you see here in a potato cell. Amylioplasts store starch. All right, so now let's talk about aspects of the cytoskeleton. So the cytoskeleton is a network of interconnected protein filaments that supports, organizes, and moves eukaryotic cells and their parts. Okay, and they play a very important role when the cell divides. So you have different types of cytoskeletal elements. You have microtubules, which are hollow filament of tubulin subunits, which are basically type of protein. All right, and microtubules are involved in movement. Then you have microfilaments. They are fibers of actin subunits, and they work to reinforce membranes, and they are also involved in muscle contractions. And then you have intermediate filaments. All right, they're stable cytoskeletal element, and they function in providing structure and support to membranes and tissues. All right, and they are the most stable parts of this cytoskeleton. Okay, so one thing before I move on, these parts of the cytoskeleton have associated motor proteins, which move cellular parts along the cytoskeleton filaments when they have energy from ATP. All right, so here are the different. So you have microfilaments, intermediate filaments, microtubules. Okay, and like I said, they have these motor proteins that are associated with these cytoskeletal elements, and they move cell parts when energized by from ATP. Okay, they can drag cellular cargo along tracks of microtubules and microfilaments. Flagella, okay, is an extension of the cell. They're whip-like structures that help propel the cell. Then you have cilia, which can also function in cellular movement, but instead of having one or a handful of flagella, okay, you'll have many more hair-like extension cilia. Okay, cilia are more, like I said, are more hair-like and can aid in movement of the cell and substances across the cellular surface. But both have an array of microtubules and what's called a 9+2 array. So 9 microtubules around the outer part of the flagella or cilia, and then 2 microtubules in the middle. In addition to flagella and cilia, some cells use pseudopods for cellular movement. All right, so you have different types of eukaryotic cells with their different types of movement. So here you have a euglena cell with a whip-like flagella. You have paramecium with hair-like cilia along the surface. You have an amoeba with pseudopods. Now you also have cilia in your body. Okay. Now these occur on cells that the cilia aren't helping the cell to move, but it's helping to move things across the cellular surface. Okay. So in your respiratory tract, many of your cells lining the interior of your respiratory tract are lined with cilia. Okay, because they're helping to move mucus across the surface of the cell so it can be moved to the throat or to the mouth for spitting. Many cells have secrete an extracellular matrix, okay, and it's a mixture of fibrous proteins and polysaccharides. And the specific composition of the extracellular matrix, as well as the function of the matrix, varies by cell. Okay, so you can, an example is in plants, they'll secrete a cuticle, maybe highly made up of waxes, which are a type of lipids, okay, to help prevent water loss. In the cell wall of plants, they'll secrete cellulose for protection, and then your bone cells secrete an extracellular matrix. All right, so here is examples of, so here's they're called osteocytes. Okay, and these are going to secrete this matrix that makes up the bone. All right, now cells need to interact with one another, and cellular junctions assist with that. Okay, in plant cells, you have plasmodesmata. All right, they extend across the cell walls. These cellular junctions can connect a cell to another or to an extracellular matrix. Okay, so here you have plasmodesmata in plant cells, all right, which allow for a connection between neighboring cells. In animal cells, you have other types of cellular interactions. You have gap junctions, which can allow movement of things from cell to cell. You have tight junctions. All right, you have tight junctions which prevent fluids from flowing between cells. All right, and then you have adhering junctions, which kind of act as basically almost like Velcro that anchor the cells to one another. Okay, so plasmodesmata in plant cells and gap junctions in animal cells have the same basic function, is that they allow for cellular communication between between neighboring cells. Tight junctions prevents anything from flowing in between the cells. Now, where do you think you might have that in your body? Well, the cells that line your stomach, okay, have tight junctions to prevent any of your stomach acid from leaking out of your stomach. All right, so you have gap junctions, all right, and then these are the tight junctions that prevent any sort of movement between the cells. And then you have adhering junctions, which kind of anchor the cells together. All right, so it's important to know some differences that we went over between animal cells and plant cells. Again, they're both eukaryotic cells, but they have some differences. Animal cells don't have plastids. Plant cells do. Now remember, those are like the chloroplasts, the chromoplasts, the leucoplasts. They're important for storage, energy storage, pigment storage, important for photosynthesis. Animal cells only have a plasma membrane. Plant cells have both a plasma membrane and a cell wall. Animal cells, they have vacuoles, but they're small. Plant cells, remember, they also have vacuoles, and they specifically have this big central vacuole that houses a lot of water. All right, and then some other another difference is the types of cellular interactions. Plant cells have those plasmodesmata. On animal cells, we talked about the tight, adhering, and gap junctions. So, what do you need to know? All right, the characteristics that all cells have in common. Okay, DNA, plasma membrane, cytoplasm. All right, and specifically with the cell membrane, we talked about fluid mosaic model. What does that mean? What are the differences between prokaryotic and eukaryotic cells? What are the differences between plant and animal cells? And then just make sure you understand those organelles, the major organelles that we talked about, and their functions. All right, and always let me know if you have questions.